All right, welcome back. It's been a while. So we got 16.7 now that we're on. 16.10 is the last section, so 7, 8, 9, 10. And I owe you guys 4 lectures. Hopefully I can get those out to you within the day. So hopefully by the end of today you have, maybe 2 days, you'll have all 4 of them, so that by the weekend you have those. And then next week, all you have to do is take the practice test and actual test, giving you a little bit of time to maybe finish some homework problems if you have to as well. But there's not a lot of them in this chapter, and so. But okay, so this lesson is about. It's one of those focus on the body lessons. I kind of like these. So we've done a lot of talk about proteins. In fact, the last lecture we did was highly focused on the structure of proteins and how the amino acid residues will or can be attracted to each other in different ways. We looked at all the different types of attractions that amino acid residues can have with each other. And structure and function, those 2 words go hand in hand in biology, or anatomy and physiology, or in this case, biochemistry. Structure and function, even in physics, whatever. Structure and function, those 2 terms go together. And so, but we're going to look at some common proteins that are in the body. So the focus is about some main proteins that are in the body that are really, really important, that get a lot of attention. In fact, one of them is the most abundant one in the body, and so that's why. But these are common proteins, and as we discuss them, we'll consider what we've learned already about their structure in terms of the amino acid attractions with each other. And so the chemistry of all that. So let's go ahead, and I'm going to switch over to my desktop here. I'm going to minimize that screen. We'll get to the lesson here. Okay, 16.7, focus on the body. So proteins, they're classified basically on how they're shaped, okay? And so if you want, you can make a major class distinction right away. And there's basically 2 types of proteins. You have fibrous, like a fiber, and you have globular proteins, like a glob, like a round glob. All right, that's the way to remember it, really. And so, and that's actually true. The fibrous proteins, they're like long fibers. They're long, linear polypeptide chains that they get bundled together. In fact, they can even do the sheets. Remember the beta sheets? So we have the alpha helix or the beta sheets in the secondary structure. They can even do that. But the point is that at the end of the day, if you were to look from a distance, the proteins are going to be like in long rods or fibers. And they're going to be insoluble. That's a key word there. Underline that or circle it, whatever. Write it down if you're taking notes. That's a huge distinction there. Fibrous proteins are insoluble. All right, they have a lot of non-polar amino acids sticking out from the fibers that make them insoluble, and in water at least. And so they're going to serve basically a structural role, kind of like the phospholipid bilayer serves a structural role because it's non-polar. Well, of course it has the polar head too, but anyways, forget I said that. The point is, the non-polar is going to be a barrier to water, and so these fibrous proteins, they're going to be giving tissues and cells a lot of strength and protection. Okay, the globular proteins, they're not so much for structure as they are for function. Well, again, structure and function always go hand in hand, but let's go back here. This is a good point to make. The fibrous proteins are all about structure. They're all about structure, and that means that their function is structure, if that makes sense. The literal structure of the fibrous proteins makes them good for structure. The structure of the globular proteins makes them good for functioning, for making them do something. So their function is to do something, like act as an enzyme or act as a key or act as a lock. We'll talk more about that when we do that lesson on enzymes. But they're going to be having a purpose. Globular proteins are interesting. They're much more exotic than your just fibrous proteins, which are just used for structure. The globular proteins, they'll get coiled and compacted into shapes with the hydrophilic outer surfaces. And that's what's cool about the globular proteins. Because they have the hydrophilic outer surfaces, it makes them soluble in water. So a lot of the globular proteins can be moved around in your blood because they're soluble, because of those hydrophilic outer surfaces. In other words, a lot of the amino acids that are in the polypeptide have hydrophilic side chains that can interact with water. And those hydrophilic side chains are usually on the outer surface of the globular protein. That's kind of the way proteins assemble themselves anyway. If you look back here at the levels of organization, we go from, the amino acid sequence to the helix or sheet, and then the tertiary structure where you finally get some 3D shape. Well, the tertiary structure, that is taking place because of all these interactions that we discussed. And these interactions basically mean that a lot of the hydrophobic stuff is going to congregate in the middle of the globular protein so they can run away from the water. Almost like a soap colloid, or a phospholipid bilayer, where all the nonpolar stuff runs and hides and meets in the middle, and then the hydrophilic stuff will be on the outside. All right, so back to our distinction here. I found something on Wikipedia you might like. I just Googled fibrous versus globular proteins, and there was all kinds of stuff here, pictures, and here's a nice little table though. Properties, we have shape, roll, solubility in water. Told you that was a big thing. Type of sequence of amino acids, its stability, and some examples. So for the fibrous proteins, they're long and narrow. Their role is structural. Remember, structural. And they're soluble. They're not soluble. Right? Your fibrous proteins, the side chains that are on the amino acids are usually non-polar, and they're usually sticking out from the rod structure, the rod fibrous structure, and then that'll make it insoluble. That'll make the whole rod kind of insoluble. And so the sequence of amino acids is usually pretty repetitive. In fact, with the examples that we'll mention, we'll talk about which amino acids you see a lot. There's just a couple of them that just keep repeating. Pretty simple, but large. There's these are really long, and they do twist and coil. So there's a lot going on here, but at the same time, pretty basic. The stability, they're definitely less sensitive to changes in heat and pH. So these guys are pretty tough. The fibrous proteins are pretty tough. Now the globular proteins, very versatile. First of all, I'm going to skip down here to the sequence of amino acids. You're going to have a very irregular amino acid sequence. So you have no idea what the kind of shape, tertiary shape, the globular proteins are going to take until you go to sequence the amino acids and really kind of analyze them, analyze all the side chain interactions. But at the end of the day, again, most of that side chains are going to be on the surface, are going to be soluble, are going to be polar, so they're going to be soluble. And the shape is going to be rounded-ish. I shouldn't say rounded-ish, but see, this is not round, but it's sort of round if you were to look from far away. Spherical. And in this case, they're going to be functional. They're going to be very functional. Basically, the shape of this protein is going to dictate what kind of job it'll do. Like, for example, there will be chemical reactions that take place with just this little mechanism right here. It'll be like a landing pad. Imagine a little helicopter flying over here and landing. That's kind of what happens with these globular proteins. Their interactions, and it's usually just one little small part of the globular protein that's interacting with the next molecule, right? But that little space or that gap that's open there, it's like a parking spot for another molecule. And so I don't want to do too much talk about enzymes yet, but enzymes that work that way with a lock and key. And so they're going to be a little more sensitive to changes in heat and pH. But again, that probably depends on the type of globular protein you're looking at. But because there's so many different types of globular proteins. Some great examples are hemoglobin, insulin, and so those are some that we'll actually look at. Let's go ahead and get right in. The keratins. Alpha keratins is the first one that we're going to look at. The alpha keratins are proteins found in your hair, your nails, your skin. Again, very structural stuff. This is all structural stuff. Your hair is not something you can't live without, right? You can cut your nails, you can cut your hair, you're fine. You need your skin. Sheeps, they can lose their wool and they'll be fine. Keratins are pretty much structural. And I was wondering, is there a beta keratin? And there is a beta keratin, so I'll bring that up in just a minute. But usually for mammals, we're gonna have the alpha keratin. Is usually what you're gonna find. And so they're composed of almost exclusively long sections of alpha helix units having large numbers of, here they are, alanine and leucine residues. Now, alanine and leucine are very nonpolar amino acids. And the alanine and leucine that are nonpolar, well, they're going to be sticking outward from the helix, meaning that that whole helix in itself is going to be nonpolar. And so it's going to be very insoluble in water. Insoluble in water. It means it won't dissolve in water, right? It's almost like oil and water. They don't mix. Neither will these substances. So what's going to happen actually is two alpha keratin helices will coil around each other to form what's called a supercoil, or sometimes called a superhelix. These in turn will actually form larger and larger bundles of fibers, ultimately forming a strand of hair, as shown in this schematic. So this is interesting. Like a strand of hair, if you just look at one strand of hair, it's pretty tiny. And to think that that tiny strand of hair is made up of just tons of these bundles of not coils, but supercoils. And these supercoils are made up of two. Each supercoil is made up of two alpha helices. Alpha helices. All right. And so if you just have one of these alpha helices, there you go. There's your there's one of the polypeptides that make up keratin. That would be your hair, skin, and nails. Now, if you're other animals, you'll have hooves instead of nails and fingernails and stuff like that. But it's the same idea. Now, this is key too here. This is key. Alpha keratins also have a large number of cysteine residues. Do you remember what we said about cysteine residues? The cysteine amino acids are very unique. If you come back up here, you'll see that it's the cysteine. Let's find those right. Where are they? Where are they? Right here. You see the disulfide bond? Well, sulfur is not in a lot of amino acids. And so it's basically cysteine is what you're looking for. And we saw that actually in this example here, didn't we, with insulin. Insulin had a cysteine sulfur bonded to another cysteine. These are called disulfide bridges or disulfide bonds. And it always occurs when you have a cysteine and a cysteine. Anywhere in the sequence, one cysteine will be bonded to another cysteine. And so it kind of begs the question, why didn't this cysteine want to bond with that one? Well, it's because this one was a little closer and this one was already bonded to that one, and so on. And so there's other reasons to why cysteine will choose another cysteine. But the point is, cysteine is always doing a disulfide bridge with another cysteine. You might say like, why didn't these two then do that? Well, because they're right next to each other. So it doesn't really work that way. It'd be easier for this one on this chain to connect with this one on the B chain, and then this one to wrap around, be a little more flexible. You can wrap, this whole chain can kind of bend over and cause that bond to occur. So, but these two are too close together. So, okay, sorry, no tangents here, promise. I'm trying to get this video done for you guys and my sake. Okay, so there we go. We got our supercoil, which is made up of two alpha helixes for keratin. And there's going to be a lot of cysteine residues, and because of that, disulfide bonds are going to be formed, and they're going to be very, very strong. Those disulfide bonds, if you remember, are covalent bonds. They're not just attractions. They're covalent bonds. There's a sharing of electrons. And so that's actually what makes claws and horns and fingernails so tough and so strong. But, here's an interesting note, too, that I found out that when you burn hair, you know that nasty smell you smell? That's actually some sulfur complexes because of all the disulfide bonds. When you burn the hair, you get chemical reactions and you get some sulfur aromatic compounds that you smell that make the burning hair smell bad. Okay, B. We only got 3 of these guys. We're already on B. Collagen. Collagen's the next structure. Now, a lot of people confuse this one or confuse it with the keratin. Like keratin and collagen are both found in your skin. And they both contribute to structure. So, what's the difference? Well, that Bible verse that talks about, 2 is strong, but 3 is even stronger. Who can break a cord? A cord made of 3 is super strong. So that's used oftentimes as an example or an illustration. When you braid rope and you see how tough it is. And so that's kind of what's going on here. In this case, you got a triple helix. Collagen, and this is the one that's the most abundant in protein in vertebrates. The most abundant. I think the internet said, Wikipedia said, always trust Wikipedia. 25% to 35% of the whole body protein content. So it is by far the most abundant. Makes up all connective type of tissues. And so you can see the red, blue, and green. Those are the three helixes that will come together to form the triple helical tropocollagen. There's actually a lot that goes into the final assembly of collagen, but what I want you to understand is that it's made up of three polypeptides that make up collagen. So collagen is like way tougher. Collagen makes up tendons and ligaments, and so it's going to be much, much tougher. Even like the gut and blood vessels. Blood vessels are actually pretty tough, believe it or not. And so the collagen is what makes it that way. It's even found in muscle tissue, in the endomysium. So if you look at a muscle tissue or a muscle fiber, there's going to be an endomysium with bundles there, and you'll see collagen there. 1 to 2% at least. Collagen constitutes 1 to 2% of muscle tissue and accounts for 6% of the weight of strong tendinous muscles. Isn't that crazy? So collagen is another good one, big one for structure. Gelatin, if you've ever heard of gelatin, is actually produced for that as well. Basically, it's made by doing an irreversible hydrolyzed form of collagen. So you hydrolyze it irreversibly, and you'll get some gelatin. A lot of people don't consume gelatin in like marshmallows or use gelatin in their Jell-O if they're vegan, because the gelatin came from ground up horse bones or horse cartilage. In fact, they even made glue from the tendons of horses back in the day, I guess. So you learn a lot on Wikipedia. You really do. I think it's better than the textbook. I was reading about how they use collagen in cosmetic surgery, bone grafts, and tissue regeneration, reconstructive surgical uses. Collagen's pretty awesome. It basically can do a lot for structure. Now, it doesn't really serve a purpose for like causing a chemical reaction to occur or anything like that. It's not really an enzyme, but it's all for structure. And this is interesting. Basically, the collagen protein is going to have glycine, proline, and then X, whatever amino acid, or glycine, X, just pick an amino acid, and then hydroxyproline. But basically, you're always going to get that kind of sequence there repeating over and over and over. So that's why if you look at the abundance of amino acid residues in like mammal skin or fish skin, glycine and proline, they're at the top. Like out of 1,000 residues, you got 300-ish, over 300 glycines. Proline's also at the top there. Same with alanine, and then of course hydroxyproline. And here's all of the amino acids that are in there. So this is not something I'm needing you to memorize, but point is you see glycine and proline a lot. And so Here you go. You see that even the book says that glycine and proline account for a large fraction of its amino acid residues. Collagen, again, is found in teeth, bone. So most of your connective tissue is going to be tough because of the collagen. And it forms kind of a weird left-handed helix, because that's usually kind of more rare. And then 3 of these helixes will wind around each other to form a right-handed helix. That's the triple right-handed super helix, or triple helix. I would say triple helix because it will help you remember the 3. You could call it super helix, but remember how this was called a super coil? It's called a super helix too. So super helix is vague. Triple helix is more detailed here, and super coil would be more detailed to describe the 2 alpha and keratin, alpha polypeptides and keratin. All right, so let's see. The side, here's an interesting part of why glycine is always found so predominantly in collagen, and it's because glycine has only 1 hydrogen atom on its side chain. All right, it's literally just a hydrogen atom. That's it. Remember, glycine is the most simple amino acid there is. So because of that, the glycine content allows the collagen superhelices to lie very close to each other. And so it stabilizes the superhelix or the triple helix via hydrogen bonding. So there's hydrogen bonding that's holding all 3 of these strands together, or the 3 polypeptide chains, I should call them. And so there you go. And now this is interesting. Again, I was saying on Wikipedia, you learn a lot. I was looking into like how this actually all comes together. And this is all I need you to know, though. At the end of the day, vitamin C is required for a lot of the reactions to occur that hold these together. Basically, one of the amino acids, if you notice, you might have saw this. Some of you may have saw this. I know you're doing your homework. You might have saw proline here, but then hydroxyproline. And you might have said, hydroxyproline wasn't one of the 20 essential amino acids in our list. And that's because it's not really. Hydroxyproline is a result of vitamin C reacting with proline. Same thing down here. There's a hydroxylysine, and that's what you get after vitamin C has reacted with standard lysine. And so if you don't have vitamin C, if you have a deficiency of vitamin C, then you won't get this fancy braided rope, right? You're going to get you just won't get that, okay? Let's just say that. And so when there's a deficiency, the collagen fibers, they don't form properly, and scurvy is what results. Scurvy is just the name of the disease. And so basically, you're going to have weakened blood vessels, because we said blood vessels are tough because of the collagen. Now, I know they're not like bone tough, but they're pretty tough. If you try to puncture it with a needle, if you've ever gotten like an injection or whatever, you'll see that like here's what amazes me: like when people stick IVs in blood vessels, and then they just leave the patient, and the patient walks around, they even move their arm and all this stuff, and I'm like, oh man, it seems like just the blood vessel's so thin that little plastic that's inside the blood vessel would just rip it or tear it so easily. But blood vessels are tough. They're very flexible, but they're tough. That's because of the collagen. And so if you don't have good collagen, your blood vessels will be weak. You'll have spongy and bloody gums, dark purple skin lesions, and there's a whole bigger list too of side effects. I saw that on Wikipedia too. So that's a big one. It made it in both articles, Wikipedia and our textbook. You got to have vitamin C, or you're gonna get scurvy because you're not getting collagen production. And we all pretty much get enough vitamin C probably in our diet. So finally, let's talk about the last one, hemoglobin and myoglobin. These are kind of like brother and sister, and these are going to be structural, or I'm sorry, what do you call them? Globular proteins. So these are the ones that are going to have some function. They're not going to be necessarily for structure. They're going to be about function. So globular proteins, remember, take on kind of a roundish shape, and hemoglobin and myoglobin are going to be conjugated proteins. What that word conjugated proteins means is that it's a protein, but it also has a non-protein molecule somewhere inside of it. And so in this case, for hemoglobin and myoglobin, that non-protein unit is called a heme. A heme. And so here it is. Here's a heme. Here heme is. And it's basically, here's the I love it. Here he is. Here heme is in the center. Here he is in the center of the heme. And he's an iron 2+ ion. The iron 2+ ion in the heme molecule is what makes this molecule special. That iron 2+ ion is what loves to attract oxygen. That's what makes oxygen stick to the heme group, because of this iron 2+ ion. And a really neat structure here too. It seems like this would be kind of a cool tattoo if you're into tattoos, or just like a cool poster or something like that. I shouldn't have said tattoos. Don't get tattoos. They mess up your collagen. So the iron, look at the structure. Isn't it cool? The iron is surrounded by cyclic nitrogen structures. You have nitrogens surrounding the iron, and it's really symmetrical, this whole thing is. That's kind of neat. But all of this is called the heme, and it fits just right here for a myoglobin, or it's right here, 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 and here for hemoglobin. And so the rest of this structure you see just in ribbon and protein ribbon diagrams, because those sequences of amino acids aren't super important. I mean, they help determine the shape of these structural proteins, but at the end of the day, it's these heme groups that are essential for the function of these globular proteins. And so again, because it helps bind to oxygen. So for us, well, we have both. All mammals have both. But for whales or elephant seals that dive down deep and they need oxygen to stay down there for long periods of time, they're using the myoglobin. And the myoglobin is found in your muscles. And so myoglobin is good for storing oxygen. But hemoglobin is way better for transporting oxygen. And so we pretty much rely on hemoglobin. And myoglobin is kind of like a last resort if you're a whale or something like that and you need to dive down. And so here's the differences between the two. Myoglobin has 153 amino acid residues in a single, underline that word, single polypeptide chain. All of this is one polypeptide chain for myoglobin. This is just one polypeptide chain. It actually has 8 separate helical sections. So you have 8 helical sections, but it's all one polypeptide chain. In other words, there's only one gene on the chromosome that codes for this entire polypeptide. Let me finish this. Myoglobin also has that heme group held in this little cavity inside the polypeptide, and it basically gives cardiac muscle the ability to hold on to oxygen. And the book says it even helps the muscle have its red color. Not that that's important, but it does give the muscle the red color. Hemoglobin consists of 4 polypeptide chains, so you actually need 4 genes. Well, you need 4 sections of DNA that will code for 4 separate polypeptide chains, and those will come together in this quaternary structure. Remember how we had the different structures of proteins? Tertiary is the shape of just the 3D shape of a polypeptide chain. But if you have a complex of polypeptide chains, like more than one, in this case, I think they're using literally hemoglobin as the example here. You've got 1, 2, 3, 4 polypeptide chains that are coming together in this quaternary structure. That's what's going on with hemoglobin. And for each polypeptide, there is a heme group. And so there's going to be more nonpolar amino acid residues than myoglobin, which means they're all going to hide down here in the middle and make a bigger structure. And when each subunit is folded, some of them remain on the surface. And the surface nonpolar subunits will actually be attracted to the other nonpolar hydrophobic groups, and that'll kind of help stabilize that whole quaternary structure. Now check this out. This is interesting. Carbon monoxide, if you've ever heard of carbon monoxide poisoning, The way it works is that the carbon monoxide will bind to the Fe2 plus ions, the iron 2 plus ions that are in the heme groups. Carbon monoxide will bond there, and if it does, it's about 200 times more strong than the oxygen would bind there. And so the iron 2 plus ion is going to hang on to the carbon monoxide 200 times stronger than the oxygen, which means, guess what? You ain't getting no oxygen taken and traveled through your blood. And so that's how you die. Hemoglobin, complexed with CO, again, cannot carry oxygen to the lungs and tissues, and without that available, cells can't function. Cells can't function. They can't do cellular respiration. They can't do the mitochondria can't do its work and make ATP, and everything shuts down. You got to have energy. It's like unplugging the computer. If you don't have ATP, your cells can't do their stuff. And so you would have other symptoms before you just keeled over and died, so don't worry. If you're around carbon monoxide, you'll probably get dizzy and nauseous. Okay, so basically the properties of all proteins, like we said, depend on three-dimensional shape. And so that is super characterized here in these images between myoglobin and hemoglobin, right? Here's another interesting disease found in the body. Remember, this is all about focus on the body, and then we'll be done here. The next lesson will be protein hydrolysis or denaturation. So if you've ever heard of sickle cell anemia, it's basically hemoglobin that is like a sickle cell. You see that little sickle shape? The sickled shape means that the red blood cell has a funny shape like this because there's a mutation in the genes. So remember how we said DNA codes for making proteins. That's the next macromolecule, by the way, that we're going to look at is DNA. And DNA, its only function is to be translated into certain polypeptide sequences or amino acid sequences. Well, if you change wherever it is on the gene, if you change the nucleotides so that the codons code for valine instead of glutamic acid, then literally just that small change will mean that one of the four polypeptides that make up the hemoglobin will be funny shaped. It won't be the right same polypeptide because you have valine instead of glutamic acid. So it's going to affect the attraction between the side chains, and the whole thing just falls apart, sort of. Which is interesting. The replacement of one here's a big deal though. You're replacing one acidic amino acid, which was glutamic acid, with a non-polar amino acid, valine. So it's going to have a pretty big change, even though it's only one amino acid. It's going to change the shape of hemoglobin, which has profound effects on its function. That means that red blood cells aren't going to be able to carry oxygen like your normal healthy red blood cells, and because they're also very fragile, and so they'll rupture, they'll occlude or clog capillaries, and they'll cause pain and inflammation, and you'll get anemia, so you'll be short of oxygen, and you'll just have organ damage, and the end result is apparently painful and a premature death. The disease is called sickle cell anemia, and it comes from a sickle-celled hemoglobin. All right. This is interesting, though. It's found almost exclusively among people originating from Central and Western Africa, where malaria is an enormous health problem. And what they found was, I guess, that the genetic mutation in the DNA is responsible for it. And so, obviously, and so that if you have that mutation in your DNA, then the parents, if both parents have it, then they're going to give it to their child, who's going to end up with that same disease. I'm sorry, I'm just trying to read this last part. Okay. That's going to be it. So there's 2 questions here. That's it to answer. Wow. The next lesson will be on protein hydrolysis and denaturation. So thank you for watching, and we'll see you on the next one.